Hello everyone, welcome to our YouTube channel Applied Forensic Research Sciences. Today we are going to discuss an instrument named Transmission Electron Microscope, which is also named as TEM. I am Anushka Mukherjee, a member of Applied Forensic Research Sciences. Transmission electron microscopy is a microscopy method in which an image is formed by passing an electron beam through a material. A suspension on a grid or an ultra-thin segment less than 100 nanometers thick are the most common specimens. The interaction of the electrons with the sample as the beam passes through the specimen creates a picture. The picture is then enlarged and focused onto an imaging device, such as a fluorescent screen, a layer of photographic film, or a sensor linked to a charge-coupled device, such as a scintillator. Because electrons have a shorter de Broglie wavelength, transmission electron microscopes can image at a far greater resolution than light microscopes. The tool can record exquisite detail as tiny as a single column of atoms, which is thousands of times smaller than a resolvable object observed under a light microscope. In the physical, chemical, and biological sciences, transmission electron microscopy is a common analytical technique. Cancer research, virology, and materials science, as well as pollution, nanotechnology, and semiconductor research, as well as other subjects including paleontology and palynology, use TEMs. Do you know? TEMs have the ability to magnify things by up to 2 million times. Consider how little a cell is to get a better picture of just how small that is. Now let's discuss about the different components of transmission electron microscope. First is electron gun. A tungsten filament or a lanthanum hexaboride, lane B6, source, known as an electron cannon, is used as an electron emission source in a TEM. When this cannon is connected to a high-voltage source of around 100 to 300 kV, it begins to spew electrons into the vacuum through thermionic or field electron emission. Then there is the cathode and the anode. Electrons leave the cathode and shield, accelerate toward the anode, and pass through the center aperture with a constant energy if the high voltage is stabilized sufficiently. The electron gun's management and alignment are vital to its successful operation. When an electric current is sent through the coils in the electromagnetic lenses, an electromagnetic field is formed between the pole pieces, causing a gap in the magnetic circuit. The strength of the field, and hence the power of the lens, may be changed by adjusting the current through the coils. The electromagnetic lens is surrounded by coils. A specimen holder holds the specimen, which is put on a mesh with a diameter of around 2.5 millimeters. Copper, molybdenum, gold, or platinum are used to make the mesh or grid. The specimen stage also has airlocks that allow the specimen holder to be inserted into the vacuum with minimum pressure rise in other parts of the microscope. A single projector lens may offer a magnification range of 5:1, and a greater range of magnifications can be produced by using interchangeable pole pieces in the projector. Modern microscopes use two projector lenses, one called the intermediate lens, to allow for a wider range of magnification and a higher overall magnification without increasing the physical length of the microscope column. For real-time imaging and modifications, TEMs originally employed a fluorescent screen that emitted light when touched by the transmitted electrons, as well as a film camera to record permanent, high-resolution pictures. Working of TEM. Electrons are emitted from an electron source at the microscope's top and pass through a vacuum in the microscope's column. The electrons are focused using magnetic lenses into a very narrow beam, which is then guided into the object of interest. After passing through the object, the electrons collide with a detector. Depending on the sample's compositional density and crystal orientation, incoming electrons are dispersed and lost from the beam in traditional bright field imaging. The intensity of unscattered electrons creates a shadow picture of the specimen, with various areas of the specimen appearing in varying shades of darkness depending on density. It is also possible to create a 3D representation of a specimen by rotating it and collecting numerous photographs at each turn, tomography. Electron diffraction can also be used to examine the crystal structure of materials with a regular atomic structure, crystalline material. Positive interference in the back focal plane produces discrete regions of electron localization, which may be seen by mapping the back focal plane to the imaging device. The diffraction patterns can then be utilized to determine the specimen's crystal structure. This is an illustration of how the electron beam originates and travels down and falls on the sample and finally shows us the magnified 3D image of that specimen. TEM can magnify any material to this much. In this image, a small fiber was viewed under TEM. See how magnified image can be available using TEM. Thank you for listening to our lecture. For more videos of different topics, subscribe to our YouTube channel.